Chapter. I'd like to welcome everyone to this session. I particularly like to thank the speakers who have recorded their presentations, as well as the panelists that have been able to come on board. We've had several conversations beforehand to discuss some pertinent topics, but obviously we want to hear from the audience who have attended to ensure that they provide some questions, and if you have specifically questions on some of the presentations, we welcome those as well. So as I said before, my name is Marco Schito. I'm the Executive Director of the Cure Drug Repurposing Collaboratory. This is a public-private partnership with the FDA. This is an FDA-funded initiative. We also partner very closely with NCATS at NIH, as well as a number of other groups and stakeholders that we work with. CDRC is part of the Critical Path Institute. We're a nonprofit organization based out of Tucson, Arizona. We've got about 15 years of experience on drug development, biomarkers, COAs, working with both pharma and industry, as well as the regulators. Today, we're really concentrating on rare diseases in oncology. So this is an exciting area that I think has a lot of potential, a lot of opportunity, and a lot of sort of growth that we can capitalize on a variety of different things, especially when it comes to drug repurposing. And that's really our effort, is really trying to figure out how do we capture real world data in order to generate hypotheses on efficacy using existing drugs for new indications. And we've done this with FDA because they do have an app called CURE. That's what the acronym for our consortia or Collaboratory is the Cure Drug Repurposing Collaboratory is that acronym that they've pulled together on that. And although it's infectious disease related at the moment, we are completely working on an app that would actually capture rare diseases and more specifically cancers. So it's a way to be able to capture clinician submitted reports, but we know that that's not always possible, and that's not always going to be the best way to move forward. I think for some of the rare diseases it may be, but there's definitely an interest and a need to look for other ways in which to capture that data, including electronic health records, which is what another project that we're just starting on. We just started earlier this year with some funding, again, through the FDA. And we hope to be able to implement that in a number of different health systems and health settings where we can use it as a leave-in infrastructure to be able to upload data, very high-level data, to be able to generate, again, those real-world data hypotheses, and if that's something to be generated, and also to utilize that infrastructure for looking at clinical trials in the future. And that will diversify our population. Oftentimes people are not really capable of participating in clinical trials because of geographic location that they're in. So those are some of the sort of the high level thoughts that we had initially coming into this. And then really it's the potential to utilize this real world evidence to supplement RCTs for regulatory consideration. That's another aspect that that we're wanting to explore further. And this is not, this can be only made possible with a collaboration that we have here today. As you see, the individuals that are leading this effort is the Oncology Centers of Excellence at FDA. So you have the regulators already here on board and willing to discuss these topics and these issues. So I'm looking forward to this discussion. I'll stop here and hand it over to both Leslie and Sonia to take it away. Thanks, everyone.
Thank you, Marco. First, of course, we'd like to welcome everybody to the first annual CDRC meeting, and hopefully there'll be many more to come in the future. As Marco said, my name is Leslie Doris, and I am actually a pediatric oncologist by training, and I am a lover of all rare tumors. Currently, I'm the acting clinical team leader for the sarcoma team at the FDA. In addition, I am the sarcoma scientific liaison for the Oncology Center of Excellence at the FDA. My co-moderator today is Dr. Sonia Singh. So Sonia, can you please introduce yourself?
Yeah, sure. Hi, everyone. Just like Leslie, I'm also a pediatric oncologist, and I'm a clinical reviewer at the FDA. I'm actually part of the team that oversees drug development programs for pediatric cancers and rare tumors. And I'm also highly interested in the potential of drug repurposing and the utility of real-world data, given the small number of patients in these disease spaces. So I'm really excited to be here, and I certainly appreciate all of our esteemed panelists for joining us today. I'm looking forward to the discussion.
Thanks, Sonia. So we are just really pleased to have the opportunity to moderate today's afternoon session on sarcoma drug repurposing. For those of you attending, if you had not had a chance to listen to the pre-recorded talks from our very distinguished presenters, I encourage you to do so, as they really are quite informative on the topics of repurposed drugs in sarcoma, the patient perspective on drug repurposing in sarcoma, the use of registries for rare tumors in propranolol and angiosarcoma. So if you have not, you may listen to them at any time after the meeting. They will still be available. Today, I'm very excited because we have an incredible group of panelists with us who I have actually had the pleasure of working with in various capacities over the last several years. I'd like now to have each panelist introduce themselves, and if they wish, perhaps give us a small summary of the highlights of the talks that they had recorded for those either who haven't had a chance to view them, and as a reminder for the rest of us, what their talks were about today. So Dr. Tapp, if you could go first, please.
Sure will. So first of all, Dr. Doros and Dr. Singh, thank you for bringing us together for this wonderful forum and topic. My name is Bill Tapp. I'm a medical oncologist at Memorial Sloan Kettering Cancer Center. I'm the chief of the Sarcoma Medical Oncology Service. And, sarcoma for us really represents a group of heterogeneous diseases. So probably, depending on which literature you read, over 100 different rare types of cancers. And one of the things I tried to highlight in my presentation is to talk about some of the challenges, but also the opportunities in figuring out how to develop drugs correctly for some of these rare subtypes. And so I'm looking forward to this discussion because I do think to do this correctly, we need a collaborative effort, not only of patients and patient support groups, but also regulators, pharmaceutical companies, and academic and physicians as well. So thank you for having me here.
Thanks, Dr. Tapp. Denise Franke, if you could please introduce yourself.
Oh, yes. Thank you, Dr. Doros and Dr. Singh, for inviting me to participate in this panel and to speak to the inclusion of the patient perspective in exploring the potential for drug repurposing in sarcoma. I'm a nurse practitioner in the sarcoma program at the University of Michigan, and I've worked with the sarcoma advocacy community in the United States to bring together various subtype-specific groups as well as broad sarcoma advocacy groups to build connections, identify commonalities and opportunities. I'm also a member of the board of directors of the Sarcoma Patient Euronet, which is an international sarcoma advocacy group. And as I mentioned in my presentation, the advocacy community is really keenly interested in drug purposing. And I kind of had gone over some of the key areas for engaging advocates to inform an initiative like this. Clearly, there is a role and an important place for the voice of the patient. And we have identified a group of sarcoma advocates that are willing to work together as a task force and really lend the patient voice to this initiative. And I'm looking forward to our discussion today. Thank you.
Thanks, Denise. And next we have Dr. Chrisanne Schultz.
Hi there. Thank you so much to the organizers and Dr. Singh and Dr. Doros for the kind invitation to talk today. As the other panelists have said, I think it's so important to think about this as a collaborative effort leading to the best outcome for every patient, every child, and every adult with both rare and common tumors. So I'm a practicing pediatric oncologist, as well as principal investigator for the International Ovarian and Testicular Stromal Tumor Registry and the International PPB Dicer-1 Registry. My talk was focused on our work at the International PPB Dicer-1 Registry. And if I was to try to sum that up in one sentence, I guess I would say it's that every journey matters, that our hope is to try to learn from every patient's journey. And I think that's really in concordance with the objectives of this conference today. Specifically, our goal is to try to optimize therapy and improve outcomes, both from a survival and a decreased late effects perspective. So through the development of novel therapeutics and fine-tuning what we have to us available in the meantime. So certainly happy to be talking with this group today, and thank you to everyone for joining.
Thanks, Dr. Schultz. And next we have Dr. Panziarka. He may be on mute. I'm not
You're
sure.
on mute, I think, Pam.
Can you hear me now?
Yes.
Sorry. Let me start again. I'm sorry about that. Thank you for the organisers for including me in this very interesting panel. I'm looking forward to it greatly. My name is Pan Panziarka. I'm Director of Drug Repurposing at a European not-for-profit cancer research foundation called the Anti-Cancer Fund. Our interest is in bringing new treatments to patients, and particularly those areas where commercial development is lacking for want of financial incentives. Our focus is increasingly on rare cancers, including sarcomas, and we're interested in how we get these treatments to patients quickly. So in my talk, I'd pick a specific example of propranolol for angiosarcoma and describe the story of how we have developed this and the steps required to actually get this finally to patients as a standard therapy.
Thanks, Dr. Panziarka. And last, we have our panelist, Dr. Breland Wilkie.
Hi, everyone. Again, I just want to echo my colleagues and thank Dr. Doros and Dr. Singh for the invitation to speak with you all today and be on this panel. So I'm a medical oncologist and a clinical trialist at the University of Colorado, and I also am a translational researcher. So because of the rarity of sarcomas and my interests, which focus on immunotherapy, I realized sort of early in my career that we needed to learn as much as we could from every patient. And so I basically spend my days wearing two hats. On one hand, I see patients and run trials and think about questions. And that's sort of where this idea of, looking at drugs that are already out there for other cancers and trying to figure out how to help my patients that maybe don't have approved options. That's certainly a lot of what I do with my day. But on the other hand, I'm in the laboratory and we're actually trying to study other drugs and try to figure out in tissue culture or mouse models, what are the next clinical trials that need to be done? What are the questions that need to be answered? And so we're actually currently running a drug screen using an FDA-approved library of over 2,300 different drugs, including non-oncologic drugs, with the goal of looking for unconventional medications that might help the immune response actually recognize sarcomas better. So I'm really excited for this discussion because I think there's so many directions we can look at these objectives, and excited to brainstorm with all of you today. Thanks again.
Thanks, everybody, for introducing yourself. We are really looking forward to a very interactive and informative session. So please, please fill up the chat box with any burning questions you have for our panelists to address. So I'd like to get started first with maybe more of a broad general question for the entire panel. And I think a lot of our panelists had mentioned this, not only in their talk, but in their introduction as well. So collaboration, that's really one of the big things that is really needed in a critical component of sarcoma drug repurposing. So how should we be pursuing more collaborative models for drug repurposing? We have academic researchers here, we have pharmaceutical companies, publicly funded research organizations, strong advocacy groups, and other stakeholders. So how can we be more collaborative in an area that is really needing our help? And this is just open, please, anybody that would like to answer.
Dr. Doros, I'll start. I think all of us who deal with rare diseases understand the need for collaboration, and we have a very collaborative international community, but it takes collaboration across disciplines, as you say, and looking at some of our most successful drug development strategies in rare diseases, it was really collaborations with pharma to help facilitate the studies and the research, the international communities to perform studies, and even patients and patient advocacy groups to understand diseases and to understand how to best improve outcomes, right? And outcomes that are meaningful for patients. And one of the things that I think we need to do is come up with a central question, an idea, and then bring together the different constituents to the table so we can all build off of each other to understand what we're trying to accomplish, and then how do we do that, right? And I think having that multidisciplinary input in the real time is what's most important.
I
Yeah.
have heard a lot of people talk about that as also being very important, is that multidisciplinary group that's really key for these type of sarcomas. Sorry, Bree.
No, please. I was just going to add on to that. So, I think if just to kind of throw out an example there. So we have a group of investigators, academic clinicians, but really a multidisciplinary group that are focused on women in sarcoma, just sort of this informal email chain. And, we bounce off cases off each other. What have you tried for this? What have you tried for this? And so there's experience there that's really, really difficult to capture. And, we've even talked about, well, how can we pool our experience? Like, how can we create some sort of infrastructure to gather this data? And I think, one thing I'd love to hear others' perspectives on is, we're siloed in some part, from our own busy lives and careers, but there's many regulations as far as sharing of data, particularly when it comes to patient data. And so the effort that it would take for us to submit data sharing agreements or run through the IRB or figure out how to pool all this together can be quite intimidating, even though those ideas are there. So, what I would wish for, and I'm really excited to hear about some of these initiatives, is how we can create a regulatorily accepted platform or a database or a gathering infrastructure so that patient experiences or clinicians' experiences or researchers' experience can somehow be compiled together to drive those questions that Dr. Tapp was referring to.
Build on what Dr. Wilkie was saying. I think that as in Dr. Tapp's talk, he talked about like the importance of the understanding natural history. And I think as we look to the patient perspective and they can be giving consent, that many of the groups are gathering information databases. They've got social media networks where they're sharing stories, exceptional responses, how they're doing, what treatment they've received. And so I think from a collaboration perspective, this is going to be, including that perspective of the patient is going to just be so critical to, especially in rare diseases and rare subtypes, really bring these communities together. Because as Dr. Doros mentioned, there really is a strong network of sarcoma advocacy groups who are really keen to have everybody in this community collaborate, because they know that they're going to be the benefactors and their family members will be the benefactors of that collaborative initiative.
Yeah, I completely agree with all of you. And I think, Dr. Wilkie really brought up something that's important, is how do we get this data not only together, but how do we use it, which I think kind of brings me to a specific question maybe for Dr. Schultz. Data is really needed for these ultra rare subtypes, and you gave an excellent talk today on the 2 registries that you run. So, both the PPB, which is a very rare form of lung cancer in kids and a genetic predisposition syndrome, as well as the ovarian and testicular stromal tumors. So, from your perspective and having had so much background in doing this and starting registries from the ground up, what do you think are the hurdles you've had to overcome in successfully curating a natural history registry for both, the PPB and DICER-1 and as well as the IOSTR?
Thanks, Leslie. That's a great question. Certainly, these are the things that keep me up at night, right? Are trying to figure out the logistics to collect all of this, and then how do we absolutely maximize the amount of data that comes from what's then been assembled? Because I think that's our solemn responsibility, to try to then use that data to its highest good. So I think to me the main question is, how do we start with the end in mind? How do we decide which data we want to collect based on how we want to use that data? And probably, as you alluded to, Bree, one of the biggest challenges is deciding whether or not you truly need PHI or not. And then who will be the submitter of that information. If you can do it without PHI, actually a lot of the regulatory hurdles come down. And so we all sort of assume that the intake form would start with name, date of birth, diagnosis date, all these pieces of PHI. But I think we should start just one step before that. We should ask ourselves if that's the data that we need. If that's the data that we need, then there will be regulatory hurdles that will flow from that, and we'll just have to contend with them and still do it anyway. If we don't need PHI, then I think actually some of those regulatory burdens are much different. The way in which that's then viewed by institutional review boards and by institutions and by regulatory agencies is different. So to me, that's one of the very central questions. And I think what helps you decide whether or not you need PHI is how long your time horizon needs to be to follow that patient. If you really need to follow them, and we feel we do in PPB, for example, we really need to follow that person throughout quite a long part of their early life's journey. We do think we need to collect PHI in order to do that, in order to get the kind of longitudinal data that we need for a rare lung cancer seen primarily in children, which in many cases is part of a cancer predisposition disorder. So for that, we need lots of longitudinal data. And for true longitudinal data collection, I think you probably do need PHI, at least in today's world. But if that's not what's needed, if you're looking at collecting data over a shorter period of time or more retrospectively, you may not need PHI, and that may bring down some of those hurdles. So to me, that's one of the very first early central questions, is to what degree do you need PHI and how would that impact your ultimate structure? But the great news is we're having this conversation that there's so many people that want to collaborate that we're trying to figure out how to make that possible now. So I feel really hopeful about that. I feel really hopeful about ways that things like the Childhood Cancer Data Initiative and other related parallel efforts are really going to make some of this streamlined at an infrastructural level, and excited to see where this goes and to help in any way I can along the way.
And I think, that's one of the central, things you bring up with PHI and, but, how can we get to the point where we can share this data across all these different stakeholders? How is it that we can do that so we can actually move forward and answer some of these questions and help the patients with, with drugs that are out there that are currently not available to them?
But
in Dr. Schultz's talk, you had brought up how that the goal can't be just to develop a registry. And that really resonated with me, because the first thing when the patient advocacy groups, when we first got together and organized, one of the first things they said, we've got to build registries, we've got to build registries. And I love that perspective of how important it is to say, what is the ultimate goal? What are we going to do with this information? And that should really inform the development.
I absolutely agree. A key aspect here is where do you want to go at the end of this? And if you're talking about taking a treatment to standard of care, then you need to be talking to regulators early on, because in a very rare cancer, a case series, a well curated case series may be enough. If you're talking about a more common rare cancer, if there is such a thing, you may be needing much more information. And unless you speak to the regulators and have an idea of what level of information you need, you're not going to be able to architect the correct solution in terms of data collection, I think.
I
That's what I was just going to mention, is that the evidentiary standards for each one of these are going to be different. And, my favorite quote whenever I talk to regulators, when I ask a question, the answer usually starts with, well, it depends. And that is the case because, you know, everything is a little bit different, and, you know, you can't make generalizations for especially rare cancers in on this. So everything has to be taken with a little bit of a grain of salt and a little bit of a, well, let's see what exactly that we want to do. But I like this conversation because really what we need to do is find out what is our end goal, what is it that we want to do, and then how do we get there, and then work backwards about getting all the information. So for example, Chris Ann, we've been talking to registries, and it's been fantastic because they have all of this information, but we don't need all of that information. You know, all we really need is, what was the diagnosis? How was the diagnosis made? How sure are we on that diagnosis? You guys know all of that information. You can combine all of that to give us one answer to that. What was the treatment, and what was the treatment history, and providing all of that. You can summarize that in a report to be able to provide why was that treatment provided. So that history will provide a little bit of that information and contextualize it. What adverse events were there? What were the outcomes? And that's where I think a lot of the people get stuck with here is, you know, what is the outcome measures that we should be talking about? And that may be something that we can also discuss. But I like this sort of fact of, you know, do we need what, you know, what is our final goal? And then what do we need to do? Let's work backwards to find out what it is, what are all the different steps to be able to do that. And the good thing is that we have the stakeholders that are interested. I haven't approached industry because I think in this case, I really want to know, you know, what the ask is for industry. And until we do this exercise of going backwards and figuring out where does industry fit in, I don't want to bring groups on board and then have them sort of twiddle their thumbs and saying, well, what do you guys want us to do? So I think we can be a little bit more prescriptive on that, but this is just this initial discussion to figure out, you know, where we are and where we want to go.
Yeah, Marco, I think that was beautifully said and summarized. And, you know, when I think of a collaboration like this, I think we could easily pick an end goal of looking at, you know, say 3 diseases with 3 different drugs, and then saying, what do we need to do in each of those diseases and drugs to move, you know, the process forward, and bringing together in each of those the stakeholders who know the disease, know the drugs, know the science. And then, as you said, what information we need to actually do that will start to work its way out. And importantly, I think there will be different challenges with each one of those subsets. So if the groups also work together, you know, we'll be gaining more experiences. And ultimately in rare diseases, what you have to do is just systematically move the bar forward and chip away at it. And I think that would be something easily done with this group, and could fairly rapidly, you know, begin to bring these drugs closer to the clinic.
Building on what, I'm sorry, go ahead.
I was going to, sorry. In some of our work at the Anti-Cancer Fund, what we've done is we've gone back through the literature, looking for repurposing candidates, and very often we find work that's really standout work, excellent publication, and then we look and say, well, what happened next? And the answer very often is nothing happened next. And the reason is, in commercial development, you've got a company there who are joining the dots, saying we need to go from here to here to here to here. And what's being missing very often in repurposing is that organization, that collaboration that does that. So very often you have academics and clinicians running really interesting trials, but they don't have the development mindset. Their focus is on their patients, and so things get published and then the work dissipates. And I think the value of a collaboration like this is to stand above that and say, okay, you've done this, but we need to plan the next step and the next step to get to the end goal. And it's really important to have that kind of big picture roadmap of where we want to go. Sorry, Denise, I cut you off there.
No, I was just going to add that some of the advocacy groups have been talking about, so is there a way to have master protocols or like kind of set some standards to build off of, where we recognize that they recognize acutely how there may be some nuances and differences amongst the subtypes, but how do we leverage some commonalities to jumpstart things and move things along? Because I think that's another thing that oftentimes we hear is there's clearly such a sense of urgency, right? And I think that's one of the things of repurposing. Does that help us move things along a bit more quickly, as opposed to some of the other kind of new compounds that are being brought forward?
And I think I like what what Bill said, and put it very, very simply, that the end goal is choosing 3 diseases, 3 drugs. And so that kind of begs a couple of questions. Is I guess for Pan, have you noticed, in the work you've done, and looking in the literature and some of your own studies, and I guess in talking about, both yourself and Bill talked about propranolol with angiosarcoma, is there, or do you see any type of bias against the non-cancer drugs in clinical oncology research versus using cancer drugs?
Absolutely. I'm a member of the National Cancer Research Institute Bone Sarcoma Group, and very involved in sarcoma studies in Europe. And we see a situation where clinicians are desperate for candidate drugs for bone sarcomas, for example, and they're only looking at drugs that are already being used for other cancers, and actually sometimes the choice is, do we go with this TKI or that TKI? Where are we going to get it for free? Which is an absolutely appalling way to select a candidate drug. I think for a lot of clinicians, the non-cancer drugs are just not on their radar at the moment, and that's something that we have to change, I think.
I kind of had to pivot back to what Marco had said. So as far as like what, how can we engage our pharmaceutical companies or partners who are running these big studies? So, one thing that I always think about when we're running these studies, we spend a tremendous amount of time collecting con-meds, so concomitant medications that patients are also taking in addition to the study therapies. And no one looks at that data except in there's some deviation question or whatever. But like if you took the 10 studies that have been done, the phase 3 studies in sarcomas, many of which have doxorubicin plus something that has turned out not to move forward. The companies have decided not to advance with that. But what if you combined all that data and you mined for responders versus non-responders, and then you pulled those con meds that are going to sit in that database for the rest of eternity and said, wow, people that did better were on some antibiotic or not, or people, again, the propranolol question. What if you look at all the angiosarcoma patients and see who was on beta blockers, even within those studies? Because the power is in numbers, right? Like those patients are already studied.
I agree completely. With angiosarcoma, we are talking about a very rare subtype of soft tissue sarcoma. The only way to do this is to look at patient registries internationally. We tried working with some of a Scandinavian country to look for data on repurposed drugs and bone sarcomas. There were so few patients that there was nothing usable there. But with more countries, then maybe that would work.
There's a problem with all rare diseases, right? It's just a numbers game, and that's what makes it even more challenging on our case.
Yeah, and even oh, go ahead, Mark.
Sorry, I was just gonna mention. I think Pan, you were talking about sort of the studies that are done in academia, and they're they've got some really good ideas and they sort of take it through, but they don't finish it, or there's what happens next kind of thing. There's always the in academia and in government, there's always this sort of tech driving force that wants to pull in pharma to be able to sort of take the initial idea and then sort of have pharma take that off. But there's also an equal issue that's happening within pharma, in that there's lots of shelved products, you know, that for one reason or another, and it could have absolutely nothing to do with the activity or the safety of the product, but it's just an issue from the company, from a business perspective, that they may not want to go into that route, and so they take that product. It's great, it's perfect, but that's not what the company is doing, and so they decide to shelve it, and it sits, you know, basically idle for years. And you have this new chemical entity, which potentially could have a huge effect, but because of a business reason, that's been deemed not to move forward. I know that there are groups like Roivant that are having those initiatives to be able to do that. That may be another opportunity where we have shelved products that could have that opportunity to explore a little bit further on, you know, things that may have been developed for one disease, but you know, actually has a great you know, effect on another disease, but it's just not within that portfolio.
And I think what Bree said, you know, sometimes we come across drugs that work just by chance.
Yeah.
And I think with all the data we do collect in clinical trials now, I mean, that is really actually an interesting way to look to see, with the numbers that you have, can you parse out anything or any of the drugs that our patients are on that could actually potentially be beneficial, synergistic. You know, I mean, that's definitely a way to go. And historically, we've definitely have seen these types of things happen, where you're treated for one thing and something else that you had that you didn't even know is also being treated. So I think that's, those are all really interesting things to think about. But I think, I guess the other part of what Bill brought up is, you pick the disease, and then how do you go about picking those drugs? And what really are the methods and the ways for us to pick those drugs that really we think we have some type of information on that would have a high impact and be that, kind of low hanging fruit. Like, if we had several drugs that we think, might be, doable in this type of situation, how would we prioritize, the drugs that we know of or have data on, either from, literature, real world, natural history studies, where, how would we go about choosing the drug we think we're really going to get the most bang for our buck?
Dr. Doris, I think there's enough opportunities out there where we have fairly robust data that we could get started, right? And I think we talk about propranolol and angiosarcoma, and there's no reason we couldn't put together, a meeting of stakeholders and, all sit in a room and come out, that day with a protocol of some sorts, goals, what are the scientific questions, the clinical trial questions, how do we understand angiosarcoma, what do we need to know about, the drug, the sequencing, the application, the study design, right? And I think there are a bunch of sarcomas like that where we can begin to think about protocols, and then ultimately it's sifting through a lot of the data, anecdotal, scientific out there, to come up with other opportunities, and I think the problem won't be finding the opportunities. I think the problem is going to be finding the bandwidth, the funding, but there'll be enough opportunities where I really do think we could get started. We spoke about mTOR inhibition in PEComas, mTOR inhibitions in epithelioid hemangioendotheliomas, interesting single drug, two diseases. How could you develop that crossover of EHEs with propranolol, right? Maybe there's some synergy there. So there's enough out there, because truthfully, in rare diseases, we're always looking for opportunities. We spend our times at phase 1 posters for that one response, or talking to our colleague in Belgium about that one anecdotal case. But bringing it together collectively, I think, becomes important, because that's when an N of 1 becomes a series, when 2 people say, oh yeah, I did the same thing. And I think that's where these group discussions really become powerful. It creates a mindset of how to do it and how to move it forward, right? So we can get away what Marco and Pam were talking about the entrapments of academia, where you spend a lot of time to do a study, but where does it go? How do we get the group and the momentum to actually finish the story?
I think a lot of that momentum comes from patient groups. So Denise, could you talk about or give examples of where patient advocacy groups really have come to the forefront to push some of these initiative and drug repurposing, get those, get that to the forefront where we're actually listening and learning and doing something about that?
Yeah, I think several of the patient advocacy groups, as I surveyed them and their understanding of drug repurposing and what they're doing in this area, many of them are collaborating with researchers around the country to identify potentially candidate drugs, as well as funding some of the in vitro, in vivo work. And so, for example, the Cordoma Foundation is one example, and I know that they work with Memorial Sloan Kettering and some of the discovery work that they're doing. And then they're currently in a situation where they've got 4 trials that are open and another one planned. And so they've got a very methodical approach, and they're providing that kind of laboratory initiative. And in speaking with Josh Summer, the leader of the Cordoma Foundation, he said one of his only constraints is not that there aren't enough candidates, there's not enough money, that they have ideas and getting and securing sufficient funding. And I think one other thing that he pointed out is, as we kind of talk about it, and I love the idea of Bill saying getting people in a room and there's probably enough information. One thing that the patient advocates always bring up, and sometimes people say, oh, we can't ask them for biospecimens. They want to provide, they recognize the importance of the correlative studies, and that should not be a barrier, thinking patients don't want to provide biospecimens, because when you're going to embark on something like this, where you maybe don't have as strong of a scientific rationale, it's going to be critical to have those samples to be able to understand if it worked or it didn't work. I think another advocacy and actually scientist advocacy collaboration, an excellent example is Corey Painter at the Broad and the work that she's done with the Count Me In. Started with the sarcoma side of it, started like with angiosarcoma and included osteosarcoma, leiomyosarcoma, partnering with like the National Leiomyosarcoma Foundation, the epithelioid hemangioendothelioma group. They've started providing samples, and I believe that Corey has now opened it up even to more broad subtypes of sarcoma. And this is really where patients provide access to their medical records, and the data is collated by people at the Broad that will pull from the medical records, as well as they'll get a tumor sample to document the so that they have that, as well as they get a saliva sample. And then they also capture real world data, patient derived, their experience. What are they experiencing from the perspective of their disease? So that's another excellent example. We have smaller groups like the Clear Cell Sarcoma Group has funded laboratory to develop cell lines, and then providing funding to do testing and try to identify, are there opportunities? So as you point out, Dr. Doris, the community of advocates is just keen on collaborating and figuring out. And I think some of the challenge has been, how do we connect in to say, what's the right thing to do? Because they're anxious, but they don't always have the best sense of direction. So I think the collaboration with the research community is critical.
So I'd like to just thread the needle on a few of these things. I think that Leslie you brought up, Denise that you brought up, that Pan you brought up. That as I start listening, everybody's coming in with these little things, and that is that so low patient numbers. It's a rare disease. Got it. Most of the drugs that we're looking at are oftentimes off patent, right? There are some that are, but for the most part, most of them are not. So drug cost is not a big issue. But the funding and the cost to take this to the finish line are huge, right? And can be many years if we're talking just about a few patients a year that can get a disease. Do they meet the entry criteria? There's all these other issues from a clinical trials perspective. What do you guys think about adaptive platform trials or basket trials to be able to maximize the costs of some of these challenges that we have? So sometimes it's a similar disease in the fact that there's a pathway that's being affected. It may be downstream, but you're treating with a drug that's higher upstream that can address some of those issues for multiple different diseases. And a basket trial may be an important aspect of this. Or you could take a look at one disease, but looking at multiple different types of drugs to be able to address, which is the best drug, and then what combinations of drugs may be synergistic or more effective than one alone. And I think that, the way that we're doing it now is one trial after another another. It just lengthens the time so long that we could probably do this much shorter time frame and less cost by doing that. And I think, what are the incentives now for industry? If we're talking about, okay, let's talk about having industry involved. If it's on patent, I think that there's definitely a discussion. But if it's off patent, it's in a generic. I don't know, really where that would lead. Obviously, I don't think that industry would be interested in that. So I think from your point, I think Pan, can you talk a little bit about the propranolol example and how were the clinical trials done with that, and any lessons learned that we could take from there?
Sure. For the propranolol work, it's important to say a lot of the initial stuff wasn't from trials. It was case series, credible, well documented, well supported case series. One of the problems with that approach was it told us there was a strong signal there, but all of the different case series they used different treatments. Some use paclitaxel, some use metronomic chemotherapy, some use doxorubicin, different stages of disease, so that there's clearly a need for clinical trials. There are currently two. Neither of them is industry supported. One in France is with metronomic chemotherapy. The one that the Anti-Cancer Fund is supporting in the Netherlands is a window of opportunity trial to assess the single agent activity of propranolol. So patients are treated with propranolol before they have surgical resection. And that came through discussions with the EMA and the MHRA. And I think that's why it's important to talk to the regulators early on. If these trials are positive, which we think they will be, we need to then discuss what happens next, and that's something that's ongoing. We're starting those discussions before the trials finish, but there is no commercial support here. This is something that's going to be funded from academia and for not for profits, I think.
And this was the similar type of discussion we had earlier today with the general rare diseases group, and that a lot of the clinical trials are actually being funded by the patient advocates. They're actually coming in and saying, what does it take to actually treat this condition? And if this is a clinical trial, then we want to do it. And they just don't have the luxury of opportunity of saying, oh, well, we didn't do this trial the right way, we have to repeat it. Well, yeah, that's not something that we need to do. We need to have those discussions with regulators so that they answer the question in the right way at the first time.
Yeah, Mark, I'd like to come back to you to the point you made about platform trials or adaptive trials. There are two things to say about that. One is that they are expensive to establish. It's much cheaper to just do one single small trial. Establishing the infrastructure to do a platform trial is more expensive. But once that's there, if you do it properly, you can dial in agents that look promising and dial out agents that are futile. In some cases, we've got a disease where there may be multiple really strong candidates. We can maximize efficiency by running those in parallel, because then you've got a single control group to compare to if we're doing randomized. So I think we need to be innovative, not just in our choice of repurposing candidates, but innovative too in how we think
Trial
about
design.
trials.
And Marco, along those lines, I mean, one of the entrapments we've always had in sarcoma is following the paths of epithelioid malignancies, because these are not epithelioid diseases and they behave differently. And we're only learning about the clinical impact of many of these diseases right now. And one of the questions really come in are where, if we did a single trial or adaptive trial design, where do the trial arms start to diverge? And what are the correct outcomes to understand efficacy? And how are we marking efficacy? You know, RECIST response rates are, you know, often really not good in sarcomas, but is it patient reported outcomes? Is it stability of a disease, improvement in symptoms? And one disease, even with the same drug, may be totally different than another disease, right? So really understanding these nuances, because as you said, sometimes we only have one opportunity to answer these questions. And really thinking about the appropriate trial design outcomes, getting in patient support and advocacies to show the importance of these outcomes. So we can also go to regulators and say, well, wait a minute, RECIST may not be what we're looking for here, right? So can we design studies that are right for the agent and the patient population so that we can move them into the clinics quicker?
The other thing I would add is that, you know, with rare diseases, I mean, you're going to capture patients with angiosarcoma that are in rural Nebraska, right? I mean, there's patients out there that don't have access to a sarcoma center where they can participate in these studies. So this is the sort of opportunity where if we're studying a drug that has an established safety profile, that a community oncologist you know, is able to feel comfortable managing and prescribing a drug that may not even be a cancer-related drug traditionally, not even a cytotoxic. You know, is there a way to design our trials to get rid of some of the expensive extra data that we don't necessarily need to collect, allow for scans and labs to be done in local centers to actually make these things possible for patients to participate in, and for, you know, for either some type of a cooperative group or an infrastructure there where you don't have to be part of a sarcoma center to participate or get your patient access to a centralized way to be part of a clinical trial.
Yeah, and that's really where I was. I initiated the conversation earlier with my introduction about electronic health records and the potential to have that as a leave-in infrastructure, so that you can have community hospitals and community groups participating in some of these, where you would just require, in the end, informed consent and randomization to be able then to take that patient and enroll in a particular trial in rural Nebraska, rather than having them say, well, no, you have to fly to New York and be there for 5 weeks, and that's all on you. So it's not feasible, and that's another sort of way that funding discussions can occur, because nobody really funds that part of it, right? So that to have your patient or patients being able to access the not only the medications, but to actually come into the clinic when they're living out of state is very difficult.
Or perhaps even to build on that when they're living outside of the country. So in rare diseases, there's all the much more motivation for us to build internationally collaborative trials, and to break down any unnecessary regulatory hurdles to participation for patients for lots of reasons, from an inclusion perspective, from a lot of different perspectives. You spoke to the rural versus urban issue, but potentially US-based versus non-US based patients, people would otherwise, like just as you said, not have the ability to travel to one of a small number of centers who might be otherwise motivated to open a complex trial. How can we break down those barriers so they can participate?
It introduces a lot of other complexities in there, Chrisanne, but yeah, I like the way that you're thinking anyway.
One of the things that Dr. Schultz had mentioned in her talk that I think resonates in sarcoma is the confirmation of diagnosis. So as we talk about this and having these trials in, just broad spread, I know that oftentimes patient advocates speak to the challenge in getting the right diagnoses. And so one would think that that's going to need to be an important, where you might want to spend some money that you'll have to for ensuring that you've got the right diagnosis, no matter where the patients are being cared for.
Thank you, Denise. We've certainly found that to be really key in PPB. Our most recent analysis shows that 21% of the cases sent to us as PPB are not PPB on central review. And in a rare tumor, that makes a big difference in data, especially if we're talking about looking at outlier analyses, at trying to find this, stellar performers in any particular outcome measure. What if that patient didn't have your disease of interest to begin with? That's a big deal when we're looking at, one or two patients had an outstanding response. We really need to be sure those patients have what they think they have. Great point. Thank you.
Chrisanne, you've had a lot of experience with this, and I know, way back when I was, doing research with you in PPB, how do you see the information that you gathered with your registry trials? How has that impacted drug development in any of the diseases that you yourself have looked at and gathered data on?
Well, I wish I had a more stellar story to tell in terms of this facilitating development of new drugs. I cannot claim that. I can't say that, thanks to our PPB research, some new drug has been developed. We're certainly trying to get there. That's certainly the focus of a lot of our efforts right now. But I think right now we're trying to learn from each and every case and trying to establish as solid a natural history data set as we can for comparison for future efforts. So I think that's where a lot of our efforts lie right now, as well as trying to get agents far enough along in the pipeline that we could really, when we meet again sometime, claim a more promising answer to that direct question. But it's a work in progress for sure, but we're trying to, just as you said, Bill, kind of get the relay baton a little further down the field as best we can at each step.
And I know we talked about different trial designs and how we can do the, how best to be able to do this. And I think, we all know, as clinicians and people that, Denise, you've run, many a SARC trial, where we've looked at sort of the, enrolling all patients with sarcoma. But now that we've had, so much advances in molecular and genetic testing, and instead of maybe enrolling many different subtypes, can we somehow subgroup those that maybe have the same type of genetic or molecular alteration? Is that a reasonable approach that we could potentially do?
Very difficult in sarcomas given the heterogeneity. I mean, even within something like Ewing's sarcoma, fusion sarcomas, we're finding that what we thought was a single disease is turning out to be a number of diseases. So I think it's much more of a challenge in the sarcoma world than in the carcinomas.
I agree too, but I think it's where we need to go, right? Because, for instance, a disease like angiosarcoma is a very heterogeneous disease. And so we struggle with that as we're thinking about immunotherapies and application and even propranolol. Other diseases, like in my talk, I spoke about PCOMA, which generally have abnormalities along the, mTOR pathway, TSC1, TSC2, but doing studies in that disease where you can actually subtype the different genomic mutations is really giving insight to drug application, right? So I think the best way to narrow down the field is important. In some diseases, it's easier than others. And a disease like angiosarcoma, it's what we're abuzz now and speaking with our community is how do we characterize the disease so we can begin to study it appropriately with a given agent, and how we characterize it for immunotherapy may be totally different than how we would characterize it for propranolol, for inclusion onto a clinical trial, right? And our understanding of responses. But I think as we get away from the all-comers trial, we can begin to ask these disease-specific questions, but still very challenging.
And Bill, that was sort of I think in your talk, you brought that up a little bit about the different mTOR inhibitors and the potential to, combine those with different immunomodulators to look at that. And then I think that at the end you asked the question, could we get industry support for a rapamycin trial, for example? I think it's a good question. And then even head-to-head sort of evaluations. That's also another good question. I don't know, I haven't had the opportunity to discuss this with industry and seeing, what they would think about that. I think that it all depends on the trial design and what the questions are. And then you're right at the end, you also asked about, should we have community support on this? I absolutely agree. I definitely think we should have the community support and getting the community input on this. And I don't know if Denise, you have any thoughts on that as well?
Well, I can say that I've had some experience in trying to secure funding for rapamycin studies, and I've not been very successful. And I can tell you it's a very expensive drug to buy as well. And so we've actually purchased it for some studies that we had run, and that is really, quite costly. I think that clearly the advocacy community, there are different size groups with different resources and funding streams. And so many of them have worked together to, whether it's like we can provide travel for people, or we can provide the biospecimen collection, or, components of it. But I think, again, it speaks to collaboration. Where are the opportunities? How much does it cost? I think sometimes that's a big black box of like uncertainty. And if people understand kind of like, well, where can they best leverage their funds that they have to really be impactful and make a difference? I think that they're keenly wanting to, right? I mean, I think that's why many advocacy groups are wanting to make things better, help move things along. And so understanding where best to do that.
So this gets into that big picture, I think that Bill and others were talking about earlier. So what is it that we're trying to achieve? And I think from this discussion on propranolol, or rapamycin, if it's an expensive drug, that obviously has implications for reimbursement. And so the potential here is that, first of all, it is on patent, it's expensive, and so would a label change allow for insurance, for example, to be able to cover that? And so maybe the juice is worth the squeeze on that effort, but for propranolol, which is a generic, maybe it's not. Maybe what we need to do is just be able to identify ways in which we can convince the community that the studies that have been done and the real world data that is available is sufficient from at least our perspective of even regulatory part of it, but not necessarily going through that whole exercise of a label change, but at least going through the review process to be able to say, yeah, that looks really good. There's no safety issues, and the efficacy is, the return on investment, the ROI, as well as the safety profile is good based on the risk benefit analysis. I don't know if Leslie or Sonia, you have any thoughts on those types of things, of either going to a label change versus not necessarily going through a label change, but having a process in place to review.
I don't know if Sonia, do you want to chime in, or I'm happy to.
Don't want to put you on the
so
spot, sorry.
No, I wanted to give her a chance to chat.
Yeah.
Yeah, sure. No, I was going to say, currently I'm not aware of any non-formal pathway to allow this, but I will say certainly from the aspects of pediatric cancers and rare tumors, both of our divisions are always more than willing to be flexible and to work with collaborators, whether it's from the clinical community side or from sponsors. So I think we acknowledge the difficulties in these disease populations, small nature, and the need to really work together. So unfortunately, I don't have a great answer at the moment, but I do think that this is something we can potentially move forward with in some sort of like pilot project sense. I don't know if you wanted to add anything, Leslie.
Yeah, and I do think, like we've always talked about with this project, one of the potential end goals is with a label change, which would certainly make it much better and easier access for our patients to be able to have. I think we probably, with this particular project have a long way to go with that, but I do think, that's an important goal to keep in mind for ease of access to our patients. And one of the other things we've sort of talked about and touched on is kind of the use of real world data and real world evidence, and how can we actually use that data as well to help us. Historically, we really haven't used that data for the drug approval process, but it's really something the FDA has been thinking quite a lot about. We have, working groups that are, specifically looking at real world data and evidence, because I think it's also one of those things that's critically important, especially with these rare tumors where we just don't have, the data, or sometimes even, the patient numbers to be able to do, the gold standard of randomized controlled trials. So, how can we incorporate that? Should we be incorporating that? How do we get to the point where we can use that data and use that data along with other, things that we've been talking about with registries and natural history studies to get information on potential drug repurposing?
So Dr. Doris, I totally agree. And Marco, when we think about the question with propranolol, right, to me, it's not expanding the label, right? This is a cheap drug. It's non-toxic. I mean, technically, we could probably add that to every patient with angiosarcoma in the clinic that we're putting on therapy. To me, the real question is, what's the correct usage, right? Who are the patients who need it, and in what context, right? And so how could we design a small clinical trial where we can begin to answer those questions, right? Where we can really begin to understand the biology. Does it have to do with, the degree of receptor on the surface of the cells? Does it have to do with a certain subtypes? Is there a genomic predisposition that could allow us to choose patients? And when we think about this, like the cost of a study of that, I think is more operational, as Dr. Wilkie was saying before, as opposed to the cost of the drug. So, how can we limit, those types of operational costs? It can often cost a few $100,000s from one institution to open up a study at another institution. Could you have two institutions run the exact same study in parallel and bring the data together? Or how do we bring the science together to understand the patient population to say, let's look into it, right? So what are the operational, logistical, and then important clinical questions that could direct the use of that drug in patients?
So Bill, then just to go off on that one step further, is that what's the next step of that? So once you get that clinical trial, to me, it's changing clinical practice if we don't need to do that, right? And then how do we do that most effectively? And that's where I'm struggling with in this case.
I think if you run the appropriate phase 2 studies, that can be clear. Again, I think angiosarcoma is a disease that can respond to chemo. So if you combine it with chemotherapy, you're not going to be able to figure out the response, right? And who and why. But we do a lot of treatment decisions based on well-designed phase 2 studies. A phase 2 study can take an FDA-approved agent and put it on the NCCN guidelines with very small patient populations, or as you said, expand the label then. So, I would be pretty convinced that with the right study, we would be able to figure out how to utilize the treatment clinically.
I agree completely with Bill on the need to identify the right patient population, the right treatment schedules, and to prove this in the trial. But for the endpoint, we want this to be standard of care for the right patients, and very often a label extension is a key part of that. Prescribers may not be willing to prescribe a drug if it's off-label. In Europe, different insurance regimes, different regulatory regimes may or may not reimburse the drug depending on whether it's on-label or off-label. So, you know, in general, I think label change is a really important thing, not for the sake of commercial exploitation of the new label, but for what the label triggers. And the label triggers things like guidelines being updated, formularies being updated. It's that kind of downstream effect that follows from the label extension that I think is important to keep in mind.
Yeah, I would agree, Pan. And I would think that is a good endpoint, to say, can you change the label and what are the different regulatory bars for an agent? I do also love the idea of consensus guidelines, right? Where,
yeah,
for instance, ESMO just had a really nice, international consensus meeting about guidelines in angiosarcoma, right? And I think there's also a level of data that could allow for the expert community to support use. And I think if we know a little bit of the appropriate indication, that becomes easier and may also be the step to, label expansion. But I agree wholeheartedly.
I'd like to ask a question to Sonia and to Dr. Doris. Is there an argument here for a public interest label extension mechanism? Because label extension generally relies on a marketing authorization holder. But if we can generate the kind of data that supports a label change, is there an argument for saying, okay, the FDA or the EMA or whoever the regulator is steps in independent of the manufacturers to force the label change effectively?
There is an ongoing. Oh, go ahead, Leslie.
No, you go. You're going to say probably what I was going to say.
This is about Project Renewal. I was going to say there is an ongoing effort between both the scientific community and the FDA, of course, to update essentially outdated labels for primarily older chemotherapy agents. And so this would be more based on, review of the scientific literature and what's actually out there in the medical literature, as opposed to requiring new data coming from, a new trial, for example. So I think there is a pathway in place that could be helpful, especially in these rare diseases. But I'd let you chime in too, Leslie. I know you wanted to say something.
Yeah, I think, that is one, one of our new projects that, we've been looking at, lately is Project Renewal, and how can we, what are the different mechanisms, especially like Sonia said, with some of these really much older drugs that we have, are there ways to change the label without having to do the rigorous phase 3 randomized control trials, that have, of course, been the historic gold standard. And that's, something that's, in the forefront of one of the goals that we're looking at is how can we make, label changes with other type of data and not just the standard, gold standard type of data that we would typically look for, which I think is, one of the other reasons we, with Project Renewal, one of the other big things, of course, that I said, is real world data, real world evidence. How can that be, utilized as well? Natural history studies, that offer us so much information. And again, literature, what can we get from the literature? Of course, I think, one of the big issues with all the different resources that we potentially have to make labeling changes is, is it the exact patient population that you're looking for? Are these different patients? What disease of stage were they at? There's a lot of confounding issues that possibly, that we have to sort of work out. So I don't think it's, prime time ready for right now, because I think there are a lot of types of kinks that we have to work on right now.
And I'll just also mention that we're tomorrow at this time there is a regulatory session, and actually Dr. Sandeep Agrawal from FDA will actually be has a presentation. So it's actually already up if you pre-recorded, if you want to listen to his presentation on Project Renewal, if you're interested in that. And then also from MHRA, Daniel O'Connor talked about the ILAP initiative at MHRA. And there'll be a number of other groups that will be, bringing their perspectives in from other regulatory agencies tomorrow. So if that's of interest, that may be something that you want to tune into.
And I also, sorry, go ahead.
I think it's also important, in discussing Project Renewal and the ways to change labels, is having group discussions like this. Where, us as regulators can bring this type of information and these concerns to the appropriate people, to have an open, discussion about this, and how can we use this? How can we keep the doors of communication open, so everybody can have some type of input, because, advocacies, pharmaceutical companies, physicians, we all have a stake in this game, and how can we use that data appropriately?
And
So,
sorry,
I was just going to ask you, Bill, and others that are more on the clinician side of the equation, is that if there is an application that is available, through the smartphone, even through that you can download on your computer, that asks, you as a clinician to say, if I've just treated a patient that, an unusual patient that wasn't responding to therapy, and I had to use a drug that didn't have that indication, so it was off-label use. How comfortable would you be in providing that sort of very high level, literally maybe a dozen questions? It's not very deep. It's really, what was the diagnosis? How was the diagnosis made? How was the patient? Why was the patient treated in a new way? What drugs were used in what combinations over what period of time? And from your personal sort of judgment, did the patient get better? Did the patient get worse? Kind of thing. Just very high level of is there something here that other people can say, well, I've tried that same drug and it actually seemed my patient also seemed to be getting better. Would that be something from in a rare disease, because there's just so few of them, that we could ask clinicians that type of question, and to basically crowdsource some of this information?
I think so, Marco. It's what we tend to do in clinical practice. We go to our international meetings, so I can sit down with Dr. Wilkie and say, I have this patient, what would you treat and what have you used? And you have to remember, most of what we do is off-label, right? It's only been in the last few years that we're getting FDA approvals, but even a lot of the chemotherapies we use are off-label. And so we do have to think about other provider types of processes other than, the FDA and regulatory processes. Like, we could think about ways to generate data that could move an approved agent onto an NCCN guideline, right? Which is often what we use, and that allows us to use it off label. But I think anecdotal responses are critical, and even anecdotal responses from patient support communities. I always have patients coming in that are saying, I was on a patient support and they use this in China, what do you think? And we've actually started some treatments and trials based on that, right? So collecting that data, curating that data, monitoring that data, and disseminating that data would be absolutely invaluable.
Yeah, the only thing I'd say is, again, you're relying on what goes in as being accurate, right? And again, I think, we do the best we can. You're not going to have the opportunity for central pathology review. So, depending on what pathologist read this diagnosis, there could be changes there. And then frankly, there's bias sometimes, right? So we may think, oh yeah, my patient totally got better, but maybe, that's because their pain got better, but their scans got worse, right? I mean, there's so much nuance to how we practice. And, again, I think we all try to be conscionable when it comes to that, but you can't control that in that sort of data set.
And I agree with you, Brulin. It's not going to be the type of data that you hang your hat on, right? It's a type of data that you kind of say, you sort of scratch your head. Is there a signal here? It looks like that there's a signal here, or wow, there's a huge signal here that looks like that we should really look into this a little bit further. It's just hypothesis generating. It's not meant to be, something that we would take to the regulators to be able to do. But it's just getting the ball started, right? It's getting our ideas in line, and I think this is also where the community can come in and from Denise, your perspective, I can see this being both from a patient perspective coming in to be able to say, I've tried these types of drugs, and especially for pediatric indications, this is where you have the parents that can also be providing some of that feedback. Although, I'm sure that every parent is going to say, yes, my kid is getting much better using this drug. But by combining both the patient or the parent as well as the clinical, you may be able to at least corroborate the findings much better. And again, just providing a little bit more power to show that potentially what we're seeing is actually true. For us to take that next step, it's a sort of thing. How do you then convince somebody to spend hundreds of thousands, if not millions of dollars on a clinical trial with the data that may be sort of like in a cell?
I think this is a great opportunity for collaboration with the patient community. I can think of, for example, in clear cell sarcoma, when that group first started forming, they felt so isolated. And then they formed this Facebook group, and patients started sharing stories and information. And actually, the woman who runs it went back and started, because she realized that people were posting, well, I tried this, or I tried this worked, this didn't work. And so even though, as you say, Marco, this might not be that you're going to hang your hat on it and take, okay, full blown, but it'll at least give some, maybe some insights or inkling into, let's try this or that, and might give some ideas for how to move forward. I'm sorry, Chrisanne, I think I stepped on your toe.
Oh, no, not at all. I think it's a great way to start a conversation, but as everyone's alluded to, it's just the beginning of that conversation. So as a treating clinician potentially entering such data, what I would be really interested to see is that I could enter the data for that patient, but then I could click a button that said, yes, I'm excited about this. I'm fired up about this, and I want to talk to the 3 other clinicians that submitted a similar data with a similar response, and really looping in those clinicians and to being thoughtful about how we would then involve patient viewpoints and patient advocacy in that as well. But as the treating clinician, now I'm in a group with 3 other physicians who also tried propranolol for their angiosarcoma patient, or whatever the example is. Tried the novel thing that because I found a literature bench report that suggested it would be a useful thing. And now I can collaborate with those 3 other physicians, let's say, to do a deeper dive into that data, to validate it on our own end, to share and discuss with those 3 or 4 other physicians, and potentially a coordinating person as well. And again, I think there's work to be done to think about then how to incorporate the patient perspective into what I'm saying here. But then use that to generate that small case series, connect those people to one another, because then they can really share that more granular data, still not PHI, but the more granular patient details that aren't going to be captured in that first look. So to me, that smartphone app, that website where you enter the data is the first step in connecting people who feel passionately about a particular angle on something to one another, so they can do the deeper dive and then present, more granular validated data for a higher level use.
I also,
sorry.
Sorry.
No, please go ahead, Pam.
I was going to say, I think it's also really important for a system like this to record those cases where it doesn't work, because the people are trying all kinds of things. Patients will try all kinds of things, and it's really important for us to find out what doesn't work. What, you know, we want not just hypotheses of what will work and what we want to explore, but also we want to know what really, you know, what's making patients feel really bad, what's not working. Otherwise, you end up with a, you know, you want to avoid the accusation that what you've got is an echo chamber of fans of drug X or drug Y.
Yeah, and this, you know, you can say the same thing about research as well, Pan. You know, I've been on several types of meetings where somebody comes up and says, "Oh, we tried this and it didn't work," and then somebody in the audience says, "Yeah, we did that experiment 20 years ago and it didn't work." Well, why didn't you publish it? You know,
Yeah.
this is the thing. It's like, you know, we've got to be able to these negative results, although they're not, you know, the big splash that everybody likes to make it, but it's what drives the progression of science. And without us having to be able to say what doesn't work is equally important as what does work.
Chrisanne, do you, just going back to that, have you actually experienced, you know, patients and their families coming up to you and saying, you know, I saw this worked, and you know, have you found that way, like other similar cases, or looked in the literature, or even within the registry, have you seen anything that's like, oh, hey, I saw, you know, this person used this or that, and wow, that's a couple of patients, I should, you know, look at that a little further?
Absolutely. In fact, the first
Yeah.
example that comes to mind for me is with a neuro-oncology patient who had a very complicated tumor just right smack dab in the middle, a very surgically inaccessible location, essentially made out of bone. And I really feel like that, happily, he continues to do well now, many years after his diagnosis. And that was really, that's the first example that comes to mind for me of absolutely a partnership. I mean, this mom scoured the internet. She talked to all the other families. She brought stacks of articles and we poured through them together. And we came to a plan that then has not only helped her child, but has now been used for several other children in a similar parallel situation. So I know everybody has their own example of that. The one that comes to mind for me first is this particular one. But yes, I think that what patients bring to the table then triggers you to think and to do the deeper dive and to say, actually, rapamycin was a part of that story and wasn't the treatment we ended up with, but one that we had explored. And to do a deeper dive into why would this mom come and put rapamycin on this table, you know, in the paper in front of me, which turned out to be absolutely brilliant. And so often I think those ideas do come from patients or from their contacts online or otherwise that then they bring to us, and we can then further explore and do that deeper dive. So lots of great examples. That's just the one that comes to mind first for me in my clinical practice. But
Yeah, and I think, you know, that's a
And
that's a great example. And I, you know, having had cancer myself, you know, we would all, you know, when I was getting my infusions, you know, we would all sit around and talk about, you know, what did you use for your mucositis? And I tried all sorts of horrible, you know, shots of straight aloe vera juice, and sucking on the ice chips when I got doxorubicin, and taking lysine, and done, you know, but we would all, you know, just all the patients in a room talk about, okay, what did you do and what did you do? But did anything ever happen with that? Did anybody ever, you know, look at that to see if it actually worked? That's just a bunch of people sitting around, you know, saying, this is what I tried, this is what I tried, you know. So I think that it, you know, it's critical to be able to get, you know, really get that type of data. And is there actually something there that does work?
mm-hmm. And then to
and I
figure out how
wanted
to
to ask
share
the
that
same
back out.
question.
Yeah.
Sorry, Chrisanne. I was just going to ask the same question to actually Bill and Breland. You know, when patients approach you for drugs for off-label use, how are you guiding them? What kind of conversations are you having, and where do you take it from there?
It's a great question and a difficult question because we do get a lot of information from our patients, right? And some are very informed and others are not. And I don't mean from what the patient brings us. It's, I think, a little bit more of the literature that surrounds it. And I think to me, it's always the dialogue and as Chrisanne said, we spend a lot of time researching what may be, you know, the off-label usage that patients bring to us to find, is it safe, is it right, you know, does it make sense? We spend a lot of time talking with our community members to also find out, depending on the types of drugs, we will elicit other experts. We have a great integrative medicine center here to also talk about the toxicities of the drug, the therapeutic windows, interactions, right? So it is a process. And I think, it's exactly what Dr. Wilkie was saying. When we collect data, it does have to be highly curated, understood, vetted, and figuring out whether or not it's appropriate. And then you also put that in the context of what are the therapies that we have, right? And what could be the responses of those? So, we still want to work with the agents that we know can be helpful. And then ultimately, it's the clinical experience, the clinical trials that are available, which can also drive things too. So it's a complicated situation in rare diseases, but there is a tremendous partnership with patients in understanding their goals and what they bring to the table. And oftentimes they can scour the literature in their disease a lot more thoroughly than we can at times.
Yeah, not much to add. I agree. I mean, I think it's a lot of education, right? Like, how is a case report different than someone's testimonial that's on an internet webpage that has absolutely no scientific connection whatsoever, And the problem is that many of these therapies, with a testimonial come with a price tag, as opposed to clinical trials or even off-label drugs that we may be able to apply for in some sort of monitored setting, So it's an educational process, but at the end of the day, we try to provide, like Dr. Tapp said, I mean, it's really, what are your standard options? What are your clinical research options that are in a controlled fashion? And then if we have none of those things, what do we know about the risks? And does it make sense? I think for any of this, you need a mechanism. You need some reason to think that there's some potential for this to help you. You need some sort of data, whether that's patient experience or, anecdotes from clinicians or observational studies or whatever it may be. But that's how we need to prioritize some of these more out of the box, non-traditional therapies to look at for patients.
I noticed, so we're actually about an hour and 36 minutes into this discussion, and it's been fascinating. The time has actually just flown by. I can't believe that we've been talking for more than an hour and a half. But maybe we can also pause here and look at some of the Q&As that are coming in. There's the first one is, how important is identifying the precise mode of action for a drug of choice? It may not be the one that you expected. And those are the type of things that, yes, some drugs have multiple modes of action, and it may have been, aspirin comes to mind, where it's an NSAID and it's for pain relief, but it's also an anticoagulant. So, there may be multiple modes that were not pursued by the pharmaceutical industry or the sponsor at the time, because that wasn't their mode of action that they were looking at, but there may be other modes of action. Any thoughts about that question?
Yeah, one thing that I always think about is nitroglycerin. Over 100 years, and there are still huge arguments about what the precise mechanism of action is, and it's changed several times. What doesn't change is the effect. So I think it's important that we are certain of the effect of the drug, and when we assess a candidate, whether it's the right candidate or not, we look at mechanism of action and the evidence for it. But I think we should be slightly more skeptical about the mechanism of action than we are about the effect. So I think the effect on the phenotype is much more important than any molecular characterization about its mechanism of action.
I would agree. Yeah, I would agree, Marco. But what I would also say is, as you do the research, it's important to try to collect the data, correlative research studies. Why? Because it may inform us about the biology of the disease, or the mechanism may inform us about resistant patterns. But I agree, right? What happens clinically is always the most important thing, but the onus is on us to try to gain a better understanding of the whys.
I agree.
Any thoughts from the regulators. Obviously, you want to know what the MOA is in order for you to be able to better understand why that drug works. But then Pan's sort of and Bill's aspect on this is that, well, if it's having a clinically positive effect as well, isn't that going to be important? So any thoughts? I guess that it's really a balance, or do you see that not as a balance at all?
I would say I think it helps for us for the reasons stated previously. It does help us to know the mechanism of action, give us a rationale for why it's working in a certain disease, for example. And obviously, if there is a risk and benefit, you're looking at the toxicities, are they off target or along those lines. But I do think if you certainly have an exceptional response, whether the mechanism is like fully elucidated or clearly elucidated, may be less important in that scenario. I don't know if you have different thoughts on that, Leslie.
No, I think, first of all, can you hear me? Because my screens are completely frozen. So.
No, we can hear you.
Okay.
You.
No, I think I agree with what everybody is saying, and especially, I definitely concur with, with what, Sonia just said about the mechanism of action and what is that? What is its importance? Does it always meet and target what we're looking at, or are there other mechanisms with which it works that could potentially work with other drugs?
And then I guess that secondary question to that is that would it give you more confidence using a drug if you really understood why it was supported by these MOAs? So, I obviously I wouldn't. I think the regulators would be as well, and that's why we want to do it. But
and I'll argue sometimes we've definitely overinterpreted mechanisms and potentially studied them in diseases we shouldn't have, right, because of preclinical data, right? So I think it's great to understand the mechanism, but it's the biology of sarcomas is exceedingly difficult. And, a lot of them, we don't have a clear understanding of their cell of origin or what's driving the disease, right? But we do see responses with therapy. So there is this level of connect to say, how do we, do the appropriate research and ask the appropriate questions with these unknowns?
Is there another question in the chat? Because mine is, it says there are two, but mine is frozen.
I was just going to mention, generic Durso also asked in the chat box, do you think generic drug makers might have more of an interest in developing a branded drug or combo?
Can that happen? So could you take a generic drug that's off patent and then relabel it as some sort of patented indication? Can that happen?
Well, a lot of times what people really like to do is to be able to take a generic drug and reformulate it. And the reformulation then creates a new IP around it. And therefore, it can go in as something that can be on a label. Not only that, but it's protected, it's IP protected, and you can get, so that is definitely a way that people are starting to take a look at things is through drug reformulation. And sometimes that's needed. It could be a drug that is used in adults, but this is for pediatrics, and you need to reformulate it because you only need, 1/100th of the dose, and it can't be in a pill form, so they reformulate it in a liquid form, kind of thing. So those are things that can help, I think. But obviously, this takes additional time to be able to do that, and additional regulatory issues and concerns. And you can't now just take something that's off the shelf and do that. Now you have to also take a look at the manufacturing and all of that. So it definitely complicates things, but I think it's from an industry perspective, they probably like that much better to be able to do. I don't know if Pan or others have other thoughts on that.
I mean, one of the other ways that we see is creating fixed dose combinations of drugs.
Mhm.
So if you've got a combination of 2 generic drugs, you can put them together in a single pill, and that gives you some IP. And if there's a medical rationale for that, that's great. I mean, I think it's unethical to reformulate just to protect the IP. But if there's a medical rationale, then I think personally, I think that's fine.
So there was some other thoughts that other people had on the previous session about you know, sort of the time frame that it takes to do this. So you know, typically we look at this and say, well, you know, if this is a repurposed drug, it should take much less time because you're not going through phase 1. You've got all the safety study and things like that. But you know, as you start taking a look, and this may be something that we'll also want to do and see how many times that's been successful to repurpose a drug in a rare cancer, number one, and then find out, you know, what was that time frame like from the beginning to end, and how much time did we really save in doing that, and how much was that final cost? Because I think that a lot of this is oftentimes the theoretical saying, oh, it should be a lot cheaper, but you know, have we really done that? Pan, have you looked at that more systematically and said and done that analysis?
There are actually our focus is on repurposing non-cancer drugs, and there are very few examples. So we don't have a big enough data set to be able to talk confidently about how long and how much. But the point that because we're dealing with old drugs, you can skip phase 1s. Actually, we curate a database of repurposing trials, active repurposing trials. I think just under 20% of them are phase 1 trials. So 1 in 5 repurposing trials are still dose finding, still safety and tolerability, even though they're old drugs. Partly it's because in oncology, you're dealing with patients who may be taking quite toxic drugs. So something that might be quite safe in someone with hypertension may not be quite so safe in someone taking cytotoxics. It may be that the dose is very different, but you know, the idea that you can always skip straight to phase 2 and phase 3, the data doesn't support that, unfortunately.
I don't know if anybody else has any other thoughts, but that was sort of my feeling as I started, you know, sort of reading a little bit into this, is that there may be some thoughts that this would be a quicker win, but I'm just wondering whether maybe it's a little bit too premature to do that. I think theoretically on paper you would think so, but in reality that may not be entirely the case all the time.
I would agree, Mark. And it also, you know, comes back to the mechanism of action question, right? You may be using a non-cancer drug for a cancer indication, but you're using a different mechanism, so it may need a higher concentration, right? And there may be a different therapeutic index achieving that concentration. So it's, you know, I think there's a lot of nuances to it.
And another sort of question that came up is payers. You know, I think that we've talked about a lot of the different groups, including industry and you know, patient groups, but we haven't really talked about the payers and the payer systems that could be used in different scenarios. So in the UK, you have a single payer system, whereas in the US you don't. So the systems are going to be different, and I think a lot of people have questions about, you know, what is the you know, it may be as it always comes down to who's going to pay for this, and if it's for patients, you know, they may not be able to afford it, and if it's the payers, it's a sort of a risk benefit again ratio of you know, how much do you pay in order to ensure that that the cost is going to be adequate, you know, for that patient moving forward.
And I think, unfortunately, for rare diseases, you know, most clinicians in their offices spend a significant amount of time on the phone with payers, again, because, you know, we cannot often generate the data that are seen in the other malignancies. So it's often explaining why we're doing and that rationale. So it is a cumbersome part of the process.
I don't know how you guys do it. And to tell you the truth, I've known several physicians that have left because of the challenges on doing that and fighting the insurance and the payers all the time. But it is a system that we have, and we've got to be able to learn how to work with them. Any thoughts about the potential of, let's say, in the U.S., looking at a fairly large payer system like the VA, for example? That may be a way in which we could, even though it's a rare disease, maybe taking a look at something like that, where you have one payer to be able to address large groups. Obviously not for pediatrics, but for older individuals that may be. But I guess the VA, you'd also have families involved there too. So I think that there may be something that I don't know, could be worth exploring. Any other thoughts on payer systems? Obviously CMS would be great to start having some conversations with. Just any other thoughts? I don't know, Pam, from your perspective in the UK.
I mean, we do have a single payer system, and I think one of the challenges, even in that system, is that we have the MHRA approving drugs, and then you have to go through a HTA process to get NICE to agree to reimburse. So I think one of the ways forward to kind of address issues early on is to have joint HTA and MHRA meetings relatively early in the process. And I know in my colleagues in the EU are also looking at this joint scientific advice meetings, where you have the payers and the regulators in the same room to discuss the development of a new therapy.
Yeah, it's a little bit more complicated in the US,
Yeah.
which I'm just starting to actually dig into myself. And I don't have all of even the processes in place, but it does come down to interacting with formularies in order to really understand whether a drug is actually put onto a formulary, and then the insurers basically go. I think there's 5 formularies that insurers can choose, but they don't have to choose all of them. They can choose some of them. Some formularies have drugs in them, some formularies don't. And that's why some insurance companies will cover drugs for a condition and some won't. It's just it's all very strange to me, and in the fact that it's not really you can have so many differences in the way that prescribers are allowed to prescribe, or that insurers will actually pay for things just based on just happenstance, how things have worked out. And so it I think requires a little bit more digging into, at least from my end, but it's definitely a complex and complicated process in the U.S. to try to figure out how that's working. I don't know if others want to chime in on that.
I can just say that the patient perspective and families is just adds another layer of distress, right? Over trying to not only figure out, is there something that's going to be helpful, but can we, are we going to have access to it? Can we get to it? But then can we even afford it? And, families talking to insurance along with clinicians. I mean, there's just a tremendous amount of time and energy, especially at a time, I think, for patients and family, that it's just so difficult to add that one more layer on top of it. And I don't have an easy answer. I'm probably saying the obvious that everybody knows, but since many of the patients who are in our session can't say anything, I figured I would at least wanted to speak that, because I do think that this comes up time and time again and is such a source of distress for people. And I don't know how we best address it. And is there a way that we can maybe have a call to action? Is there a way that advocates could do something that would be helpful to change the situation?
I'd echo that. I mean, I drop into a number of patient forums with large numbers of patients from the US, and I would say that the number one question that comes up is what's, which insurer, where do I go? It's the topic that crops up again and again for all kinds of cancers, not just sarcomas, but all kinds of cancers from patients. So I think it's definitely an issue that really does need addressing from the patient perspective.
And these are sort of the issues that come from the legislative side, because I think that at some point, I think others have said this in the chat, is that we need a system to be able to move this along. And I think the Best Practices in Children's Act was a prime example where you actually had groups coming in and pressuring government to be able to say, we need a new system to be able to address this high unmet need. And I think that that's where patient advocates really come into play. They have a huge voice, and I think we just need to be able to, like a lens, be able to focus that in the right direction, in the right time, to be able to apply the right pressure to potentially make a difference.
Well, I think, thank you all for the lively discussion. We have about just a few minutes left, and I wondered if each of the panelists might, in a minute time, give us a take-home point from this discussion.
I'll jump in and start. I think the take home is that the patients have a very important perspective and an important voice that needs to be incorporated and infused into this repurposing, because I think they're powerful and strong, and they have a great deal invested in this personally. And I think that they can add and help us move this forward.
Thanks, Denise.
I'll jump in for next. I'll say start with the end in mind, that as we think about the concrete steps that we want to take, I don't think we should just think about creating the largest possible resource we could ever use. I think that's too heavy a lift for where we are right at this moment. And so I think we should think about what our true end objectives are, and then work backwards from that, and start with the most feasible way to then take those first steps toward that very specific end goal. But I think the clearer we can get about the end goal and what success would look like, the more we're likely to be able to really come up with some tractable first steps.
Yeah, Chrisanne, I'll echo that, and also say that to be able to put this into a roadmap, so that, yes, we start with that end goal in mind, start working backwards, and then develop that roadmap so that we have a path moving forward. And we know, because at some points in that roadmap, this is where industry would come in. This is where the patients would come in. This is where, that's when we can really start identifying areas where we can leverage the relationships and the partnerships that we're building.
I agree. I think we all want the same thing. And I think the biggest thing that we can focus on is how to break down the barriers that keep all of these people with all of their different interests from working together for the same goal.
I think I'm suffering a massive case of confirmation bias. I came into this meeting thinking that repurposing was had a huge potential. We just need to get everybody on the same page. And I'm going away from this meeting glad that so many of us are on the same page, and we're all kind of thinking the same kind of thing. So I think it's been a really positive meeting.
And I'll just echo that. I mean, I think we should all be empowered by this collective. It's exactly who we need at the table. I would say, let's come up with one concrete example, set the goals, roll up our sleeves, test the waters, figure out what works, figure out what doesn't, and, let's get started.
Thank you all so much. We appreciate you all taking the time out of your busy schedules to come and talk about something that we think obviously is extremely important, and especially identifying the sarcoma community, which is a small, tight-knit group that, is just a really powerful force when we can all come together. And especially to the CDRC that put so much work and effort into, allowing us all to come together and have this great discussion. And I think we now have many points that we can move forward with and kind of a plan of where to start and really make this successful.
And one thing I'll just say right at the end is that we do have work groups that have been starting to be formed around this. So if you're in the audience and you find this fascinating and interesting and want to participate, please let me know. You can email us at cdrc@cpath.org. I put that into the chat, and just email us, and we'll get you situated and have you come on board and see if we can work together. So looking forward. Many more discussions to come.
Yes, thank you all so much for a great session.
Thank you.
Thanks, everyone.
Thanks.